In this video, we're going to talk about nucleic acid metabolism, and we're going to focus mostly on their biosynthesis because they have this beautiful branched biosynthetic pathway that helps us think about how cells build diverse molecules from sort of generic starting points. So let's launch into it. Nucleic acid metabolism. So quick reminder, if we think about the set of biomolecules that we have, they range from fats and glucose, or this is a glucose, but carbohydrates as a class. They're basically carbons, hydrogens, and oxygens. As you get more complicated, you get into amino acids. Now you've added amines. And then when you get up here to nucleic acids, now we're talking about basically carbons, hydrogens, nitrogens, phosphorus. It's a much more complicated molecule in some ways. And so what we're going to be asking today is how these things get made. Other quick reminder is we've got two basic classes of nucleic acids, bases. We have the purines at the top, and these purines are, this is an adenine, and this is a guanine. And then these pair with other molecules. So this is sorry, I'm doing this wrong. This is a C here, a U here, and a T here. So a cytosine, uracil, and a thymine. So these are the 5 basic bases that we build. We also have to worry about whether we're in RNA or DNA, and that depends on the carbons or the hydroxyl state of the ribose sugar. And so what we're going to be asking today is how do cells build this complicated set of molecules, right? RNA, DNA, purines, and pyrimidines. The basic starting point, I'm going to hide for a minute here. The basic starting point for this is this molecule here, R5P. Now, R5P, if you've seen my video on the pentose phosphate pathway, that might be ringing a bell. This is one of the primary outputs of the pentose phosphate pathway. So if you're, and that's this pathway that's right next to glycolysis and gluconeogenesis. Basically take those central carbon intermediates like fructose 6-phosphate, glucose 6-phosphate. You can flip them over to make R5P, which then goes on to be the backbone for basically all nucleic acids. What we're going to do with R5P is we are going to make it into this molecule, PRPP. And you'll notice there's more phosphates on, so we've invested in ATP to do this. We are then going to do one of 2 basic routes. We are either going to build this molecule orotate and then glue it onto a PRPP, and that is going to make our pyrimidine bases. Or we're going to take our PRPP and we're going to build a molecule called IMP on top. And then we are going to use that to build to our purine bases. So this is, at the very highest level, this is the big hierarchy, is we all start at R5P, we go to PRPP, and then this is our first branch. We go to IMP or orotate, and then from there we can go into the different bases. So we can go into A, we can go into G, we can go into U, we can go, sorry, I totally screwing that up. We can go into U, or we can go into C. Here's the same information I just showed you. Well, a lot more information on top, actually. We're showing what sorts of stuff is getting put in. And so we have our glucose 6-phosphate up here, very central carbon intermediate. This pentose phosphate pathway, this complicated network of reactions that ultimately gives us R5P. And then PRPP, that is at the center of everything. From this point, what I'd like to notice here is just all of the stuff that's getting invested, right? We're investing ATPs, we're investing amino acids, we're investing formates, we're investing NADHs, we're investing, that one's actually coming out. We're investing NAD+, we're investing Q, right? So there's this big complicated network that's going to lead to these basic outcomes of our uracil monophosphate, our adenosine monophosphate, our guanosine monophosphate. We're going to dig into those as we go on here. So the questions we're going to be asking to help organize this are, where do we get these base ring structures? How do we regulate the pathway? Then we're going to go into the details, because I think it is cool to at least sometimes to dig in and just say, here's all some of the guts of this, but how do we get to RNAs? How do we get to DNAs? And then I just felt like couldn't leave this without at least saying one slide on how these amino acids or nucleic acids are broken down. So that'll be a very quick end slide there. But okay, so where do we get these base ring structures? Orotate is the core of not all 5 bases. Wow, my past self utterly failed to write this here, of all pyrimidine bases. Sorry. Typo failure. I'm glad I caught it while I was talking. And here's ortate. And hopefully you look at it and you're like, yeah, that kind of looks like a pyrimidine base. And it's built out of stuff. So let's actually look at the stuff that goes into it. So here's our ortate. And if we draw a circle around this aspartate here, you can actually see, hey, look, it is just right here in the base, in the ortate. So everything except for this one oxygen ends up just hanging out in the base. This glutamine here is going to give us the nitrogen. And then this HCO3 is going to give us the hydroxyl. So we can build this base by basically gluing together some parts, right? Gluing together some amino acid parts and some small molecule parts, but we end up with this ring structure here. If you take that orotate and then you glue it onto a PRPP, so this is our PRPP, coming out of R5P, coming out of the pentose phosphate pathway. This is our orotate we just made. If you glue them together, you get uracil, our first base. And actually, this is in what's called the monophosphate form. So this is uracil monophosphate, or UMP. Cool. Remember, this is this branched pathway where PRPP is at the center. You can do the same basic story for IMP, but if you look at that, you're like, oh my goodness, there's a lot more going on, and there's a lot more steps I'm skipping over here. But if you look into this, you can say, oh, like, what are the parts? Well, we've got our, we've got this amine is coming from an aspartate, and these amides are coming from glutamine, and you can walk through and you can trace out where all the parts come from. And what this is actually showing is a general principle, and that general principle is that cells build complicated molecules by basically gluing together amino acids in a few moieties. So you looked at both of these, you can say, oh, well, that's the glutamine and the aspartate that we showed for orotate getting glued together. It's just those amino acids that are being pasted into different places. And when you go into most biosynthetic pathways, it's going to be this principle just repeated over and over and over, because there's really not that much chemistry, right? I mean, cells do an amazing amount of chemistry, but they don't have that many parts to start from, right? They've got the CHNOPS atoms. They've got a fairly small, limited palette of reactions. It's just repeating them in different ways, and combinatorial ways lets them build diversity, build, in this case, a bunch of bases, nucleic acid bases, out of a small number of amino acid parts. Okay, so that's the general idea. That's where the ring structures come from. Now let's talk about how these pathways are regulated before we dig into the guts of how we get to these individual types of molecules. So how are these pathways regulated? This is a good chance, if you'd like, to pause and think about things for a second and ask, what do you observe about the regulatory structure of these pathways? So now I've taken the graph I showed before, the chart, and I just added these regulatory arrows on top of it. So maybe pause and think about that for a second. What sorts of regulation is going on here? All right, so came out of our pause, and what we see here is we see lots of inhibition. There are lots of points on here where, if you look at this, for example, we have this flat arrow. That is saying that AMP is inhibiting its formation. We see that GMP is inhibiting its formation. We see that UMP is inhibiting its formation. And it's even inhibiting up here, all the way up here before making orotate. And finally, we see that ADP is inhibiting the formation of PRPP. So we have this structure where there's a primary role for inhibition. We're going to dig into this, into these specific regulatory steps now, one by one. So first, let's talk about this upper step. This is right here, R5P going to PRPP. It turns out this is the primary point of regulation for all nucleotide synthesis. And that's because this right here is the shared intermediate of all things. And so you could call this a committed step to biosynthesis. Right? R5P here, I don't know if you remember this or you've seen the pentose phosphate pathway before. This molecule is part of the pentose phosphate pathway. It's a dynamic network. It can flip around. It can go to all kinds of different metabolic fates. But once you're like, nope, I'm going to invest in ATP and I'm going to make a PRPP, now you're on your way to nucleotide synthesis. So this is a primary point of regulation for that, because if you can stop this, you can basically stop nucleic acid synthesis. It's inhibited by ADP. And what does ADP do? ADP is indicating that the cell's energy state is low, right? If you start seeing ADP concentrations going up, that's saying you're using lots of ATP, which that makes sense, right? You're like, do I want to make a bunch of energetically expensive nucleic acids when my energy state is low? The answer is no. So the low energy state ADP says stop making a bunch of nucleic acids. If we look at the rest of the pattern here, what we see is that AMP, GMP, and UMP are all regulating their own production, right? They're just regulating something upstream. And that, let's look at GMP as sort of just an example. If GMP concentration is going up, it's saying, man, it's not being used as quickly as we're making it, so you better slow down making more of it, right? It's the idea of this, it's actually got a name. This is feedback inhibition. You're saying, I'm building up stuff, slow down making more of this stuff. And this actually gives us an interesting general principle, or a couple of general principles in the architecture of metabolic systems like this. First, when you think about whole modules of shared pathways, so like all nucleic acid synthesis, that's often regulated by a marker of total energy state. So ADP in this case is saying energy is low, stop making nucleic acids. Whereas specific product production is often regulated by the product. It's basically saying we have enough of the product, stop making more. Or if the product starts being depleted, then it'll start turning up that pathway. You relieve the inhibition. And so overall, we see this a lot where, again, the total energy state's regulating just are we making stuff or not? And then specific products are regulated by themselves. Okay, so we've talked about where we got the base ring structures from, sort of gluing together amino acids, basically. We talked about the regulation of these pathways. Now let's talk about how we actually get to the specific DNTPs. And this is mostly just, this is cool that we can do this hierarchically. So first of all, make myself go away again. RNA triphosphates can be readily derived from these monophosphates. So AMP goes to ADP and ATP. And we've actually seen this before, if you've been in these videos for a while, when we talked about cell energy state. Remember, cells, they can take two ADPs and they can glue them together to make an AMP and an ATP. We're seeing the same thing here, right? What we're actually doing is we are taking an AMP and an ATP, and now we're making an ADP. And then we take that ADP and another ATP, and we can swap to make ATP and ADP. So basically, once you have a monophosphate, the actual base part can be swapped around with the phosphates sort of willy-nilly. So you can readily exchange between monophosphates, diphosphates, and triphosphates. You're never going to add extra phosphotoester bonds, right? That's going to take something like glycolysis or oxidative phosphorylation, but you can swap the bonds around all you want, basically. And it turns out you can do that for AMP, GMP, and UMP without any interesting chemistry at all, just an enzymes that speed up this reaction. And then to get to CTP, right, so we can get all of our RNA bases, so we can get our A, our G, our U, and our C, we do actually have to invest in ATP and a glutamine to basically convert this. We're basically doing, it's actually not a transamination reaction because it's not a keto acid, but it looks a lot like a transamination reaction, right? We swapped out a carbonyl for an amine. Okay, so we hopefully that you're satisfied now. We know how we can go from PRPP all the way down to AMP, GMP, and UMP, and now we know how we can get to ATP, GTP, UTP, and CTP. What about DNA bases, right? How do we get to those? So quick reminder, the difference between a DNA and an RNA, it comes down to this hydroxyl group here. RNA has it, DNA does not, because the DNA stands for deoxyribonucleic acid, and this RNA stands for ribonucleic acid. So we have to somehow whack that off, and it turns out that the enzyme that does so actually only operates on the phosphate form, so the ADP, the GDP, for example. But basically, this is the deoxy reaction here. But once we've done that, once we've done an ADP to make a deoxy ADP, we can now make a deoxy ATP. So this is now a DNA base, or it was an RNA base up here. And we can do the same thing for GDP. We can do the same thing for CTP. Awesome. So that wasn't too hard, right? So we've made DATP, DGP, DP, and DCTP. What do we do to make TTP? Wow. To make the last DNA base. So you might recall that RNA uses uracil and DNA uses thymine. How do we get there? Well, it turns out it's kind of a pain, but we had to mention it because there's a cool name, cool intermediate in the middle. So we take a UDP, uracil diphosphate. We make deoxyuracil diphosphate, which is not biological, right? We don't really use a deoxy nucleic acid in DNA. So then we take that and we turn it into a DUTP. So now we have a triphosphate form of the deoxy-U form. And then we make one of the cooler named molecules. We make a dump. We basically make a monophosphate form. And then that monophosphate form can be used to make thymine monophosphate, and then we can get the triphosphate. And so basically all that we're doing there is we're putting on a methyl here, and that's coming from this thioflavin backbone. Doesn't matter. Details. We're in so many details here. But the idea is that you can do this amazing block of chemistry, right? You can start up here with glucose 6-phosphate, and then you can make a ribose 5-phosphate with the pentose phosphate pathway. So that's all sort of central carbon stuff. You can make a PRPP. You've activated that ribose 5-phosphate, and now it's ready to rock and roll, ready to go on and make nucleic acids. If a cell glues an orotate onto that, you get this UMP, uracil monophosphate. If the PRPP goes on and has the base built on it, it can become inosine monophosphate, IMP, which then immediately can be turned into AMP and GMP. And then from there, there's just simple branching pattern. I mean, it's a complicated feeling, right? But at the end of the day, you can kind of trace it out. You're like, look, I made deoxy ATP. Look, I made deoxy GTP. Look, I made, okay, this one does look complicated. Look, I made a CTP. And oh, look, I made DCPT. DCTP, wow. And then, okay, yeah, that's complicated. I agree. There's all kinds of steps that happen to get there. But then you get the deoxy thymine TP. Okay, cool. So overall, big picture idea, nucleotides are assembled in this hierarchical fashion. Last thing I just want to mention, because I felt like I couldn't not mention this in this whole slide, is how do we break these down? One slide, simple thing here. But at the end of the day, we have a ribose, right? That's a ribose sugar. That's the, do I have a picture? It's going to go too far back. I don't have a picture. But that's the 5-carbon sugar that is like the basis of these nucleic acids. That 5-carbon sugar gets recovered as ribose 1-phosphate. And so now we have made an R1P. And then we have basically have the nucleotides split off. So basically we have the base separated from the sugar. And then there are two basic possibilities. You either go into what's called a salvage pathway, which is keep using that purine for something else. Or you take the purine and you make it into uric acid. Pyrimidines, you either salvage and use that purine for something else. That pyrimidine for something else. Or you go into malonyl-CoA. You might remember malonyl-CoA from fatty acid synthesis. Same molecule. Biology reuses the same things over and over. But overall, those are the basic outcomes. So you're going to capture the sugar as a sugar. It's a carbohydrate that can go into central carbon metabolism. You're going to salvage the base. You're going to try to use it for something later. Or you're going to put it into these final outcome intermediates. Okay, that was a lot. There's a lot of details of pathways in there. But hopefully you got a flavor now for how nucleic acid biosynthesis in particular works. Basically, all nucleic acids are synthesized by a small number of branching pathways. They use amino acids and single carbon units like formate as inputs. The pathways are regulated by both cellular energy level That's the whole pathway is regulated by, do we have enough energy to make nucleic acids right now? And then by feedback inhibition, are each of the products, are they accumulating or are they being depleted? And then finally, after, there's all this branching that I'm skipping over, but I'll just mention that nucleic acids, or we mentioned at the very end, that nucleic acids are catabolized by similar branching pathways, kind of run backwards in some sense. And what this does is it releases ribosugar, uric acid, or malonyl-CoA. Okay, so that's what I have on nucleic acid metabolism. There's a lot more to be said, so look for future videos, maybe looking into that. But until then, thank you so much for your time, and I hope you enjoyed the video.